One is the mucin secreted by the lining cells, the simple columnar cells of the lining epithelium of stomach, passes into the lumen in exchange of bicarbonate. Bicarbonate moves out of the lumen into blood, and to maintain this neutrality, electrical neutrality, it has some stimulatory effect over the stomach, increases the secretion, increases motility, and some trophic effect also. Most of the questions are coming from these sort of topics. So first, about the gastric secretion. Now we all know that gastric secretion, if you think this is the stomach, functionally we can divide the stomach into two parts. One is fundus plus body. This fundus plus body produce a large number of cells that contain parietal cells. Large number of glands contain parietal cells, and these parietal cells are the source of some important constituents of gastric juice, namely first hydrochloric acid, intrinsic factor of Castle, and some cations, sodium, potassium, magnesium, calcium, all are released from these parietal cells. Secondly, they contain some peptic chief cells. Okay, this can release pepsinogen. It is an inactive form of the active enzyme pepsin, which is the first enzyme for protein digestion, first proteolytic enzyme. Okay. Besides those, there is another cell known as the mucous neck cells. They secrete mucin. Okay, look, there are two types of mucins in stomach. One is the mucin secreted by the lining cells, the simple columnar cells of the lining epithelium of stomach, and those are that mucin is responsible for the protective layer or the protective function against this hydrochloric acid. Okay, but what does this mucin do? This mucin is for lubricating action. Okay, for good mixture of this chyme, what we call the foods inside the stomach, for good mixture and thorough metabolism or digestion, this mucin is needed. Okay. In this pyloric part, there is a scanty, seldom any parietal cells for a cell secretion. Rather, this part is rich in a cell known as G cell. This G cell produce gastrin. G cells produce gastrin, a local hormone of GI tract. And there are some other cells known as the ECL cell, enterochromaffin-like cells. These are some neuroendocrine cells, and they release histamine. This histamine acts as paracrine manner. We know as autocrine, paracrine, endocrine. Autocrine means a substance secreted from this one cell and acts on that very cell. Paracrine means secreted very nearby from very nearby source and act very nearly. Suppose this local hormones act in paracrine manner. This gastrin acts secreted from G cell and act over the parietal cell. So all the local hormones of GI tract act in paracrine manners. Now, what does they do, or what do they do? If we think this one is the parietal cell, okay, this one is parietal cell. This is the luminal surface, and this is the interstitial fluid. Now, in this aspect facing the interstitial fluid, there are three receptors on the parietal cell. One is acetylcholine receptor for acetylcholine, and it responds to vagus, that is parasympathetic stimulation. Another receptor is known as the H2 receptor for histamine. Okay, this is for histamine. That histamine is released from enterochromaffin-like cells. And third one, it is the gastrin receptor, and it binds with the gastrin released from G cells. Okay, these three types of receptors act combinedly for production of the hydrochloric acid. Carbon dioxide mixes with blood, combines with blood to form a very weak acid called carbonic acid by the enzyme carbonic anhydrase, and this acid is very unstable. Okay, and they release bicarbonate and hydrogen ion. This hydrogen ion gets out of the parietal cell inside the lumen by a counter transport in exchange of potassium, and this is a primary active transport. because they need energy, active expenditure of energy. There is an ATPase enzyme acting here. So this hydrogen potassium ATPase is known as none other than proton pump. There is a large number, very popular group of drug known as the PPI, proton pump inhibitors, that act here, and chloride then passes into the lumen in exchange of bicarbonate. Bicarbonate moves out of the lumen into blood, and to maintain this neutrality, electrical neutrality, large amount of chloride is passed into the lumen from the cell. Okay. So this hydrogen ion and chloride in turn combines to form the hydrochloric acid. So this is the mechanism of secretion of hydrochloric acid in a very nutshell. Okay. Now this vagus not only acts over this receptor, moreover they stimulate, this vagus stimulates both ECL cells and G cells to release their secretions. That is why when we perform vagotomy, truncal vagotomy, in case of our any form of vagotomy, in case of pyloric stenosis or long standing duodenal ulcer, not only this receptor is blocked, but this effect is also blocked over the ECL cell and gastric cell. So their secretions, though not completely abolished, but reduced to a greater extent. Okay. So this is the simple mechanism of gastric acid secretion. Now there are a number of factors that stimulate this gastric secretion, and there are factors they inhibit the gastric secretion. With time we will get to know about this. So now there are three phases. Okay, there are three phases of this gastric juice secretion. First phase we call this as cephalic or cephalic phase. It occurs even before we even before the food enters into the stomach, and it can start with smell of food, hearing about the food, sight of the food, or even food in the oral cavity. And this phase is mediated by the vagus.